that better? So I'm Matthew. I've been a professor here at MSU for almost 14 years, maybe 15 years. Microbiologist, environmental microbiologist. I did my doctorate at Cornell University in microbiology and bioengineering. Had a faculty position, well, I did a postdoc at Oak Ridge National Laboratory. It's in Tennessee, a Department of Energy facility, and I still do some work related to the Department of Energy. And as I said, I've been here for not quite 15 years. I'm a professor in microbiology, and I'm also director for the Center for Biofilm Engineering, which is a research center here on campus. It's actually celebrating its 30th anniversary. So it's one of the longest lived research centers that came out of a funding program of NSF and that we're still going today. Is there a remote? No. As I said, I'm an environmental microbiologist. I do quite a bit of different things in my lab. We study sulfate-reducing bacteria in the context of linking carbon and sulfur cycles for a lot of environments. One area, though, that this is a very applied area that has repercussions is that sulfate reducers impact metal corrosion. So this is big in the oil and gas industry. In fact, we have a project right now with Equinor, which is the Norwegian state-owned oil company, where we're trying to help them solve issues with corrosion of pipelines, oil pipes. And this is then why we have environmental catastrophes and we get pipelines break and then you get oil leakage. And this is really being carried out in a big way by microorganisms and the biofilms that form on the inside of those pipes. So that's one group of organisms that I study. We also study methanogens and methanotrophs. These are organisms that either make methane or eat methane. And this is obviously going to be important for the carbon cycle. Obviously, too, with biofuel for the potential of using natural gas or methane. These organisms also have a lot of unique biochemistry in how they process C1 compounds because they're interacting with CO2 and methane. These are both C1 compounds. So they have a lot of interesting biochemistry to understand how biology processes carbon molecules like that. I study phototrophic eukarya. This is algae and diatoms. We study them in the context, obviously, of fixing carbon. So this is also important for the carbon cycle. But as you'll see, we look at them as well for biofuel production. They make lipids and oils. You can turn this into biodiesel. So we just got a new grant funded by Department of Energy this summer to further understand algae and diatoms and their ability to make lipids and how to turn that into fuel. And then for a lot of these environments, I also study microbial ecology. In my postdoc at Oak Ridge, I worked with a microbial ecologist, and we're interested in the microbial communities that are associated to a lot of these environments. And how do we understand the structure and function of a microbial community? And what I mean by that is how the microbial community is structured, what populations are there, how those populations interacting, and ultimately how that impacts their function, what they're doing. Along in those lines, I have also a new project in 3D printing where we're looking at how to 3D print biofilms. That's actually funded by the Department of Defense, where we can we start to print biofilms in certain structures or patterns to get them to do things for us. For instance, processing carbon. So right now, my lab's funded by the Department of Defense, the National Science Foundation, and the Department of Energy. Next slide. So again, I mentioned real quick this notion of structure function, and a big question in my lab for all of these environments is there a relationship between the structure and the function in that microbial community. And it doesn't matter whether you're going to think about the human microbiome, and I know you've had some folks talk to you this semester about human microbiomes. It's a big area in microbial ecology right now. These are the exact same questions, whether we're thinking about the gut microbiome or I'm going as an environmental microbiologist to dirt. It's really the same questions. And can we understand the causal basis? That's not casual, that's causal. Can we understand a causal basis, meaning cause and effect? Meaning when you got this is what's going to happen. We don't really have a really good understanding for many microbial communities of all of how that happens. And we want to understand a causal basis for how microorganisms assemble themselves and organize themselves and have all the activities that they have across a given environment. Next slide. So next slide. One of the first areas is heavy metals, bioremediation that we study. And so I'm concerned with, why do we care about this? Why does DOE and DOD and NSF give me funding for this? Is to keep water clean. And if you don't, a lot of people appreciate the fact, obviously, that over almost 3/4 of the Earth's surface is covered by water. But when you start to think actually about drinkable water, where is that? And most of that, a vast majority, 95% of Earth's consumable and available water is groundwater, meaning it's in the ground, meaning it's below the surface. This is meeting, all this water is meeting supplies for irrigation and industrial needs, and obviously drinking water. Groundwater supplies 3/4 of the drinking water in Europe, 1/3 of the global population, and when you think about the United States, over half of the US population, drinking water source is groundwater. I live a couple miles out of town. I'm not on Bozeman City Water. I have a well. 51% of the population, 99% of the US population, rural population, is on groundwater. This is a huge resource that we're not taking very good care of. Next slide. This is a USGS map looking at aquifers in the United States. This is water below the surface. This is by definition why we call it groundwater. It's in the ground. It's not the water that you see. Look in comparison to the Great Lakes. We always think of the Great Lakes as this huge freshwater source, vastly diminished in comparison to the underground aquifers that we have in the United States. Next slide. And this is just real quick looking at a water balance from a reference down below. And this groundwater has a huge surface area compared even to the oceans, and it has a very long residence time. Residence time meaning how long is it in the ground before it comes back up. And it can be, sometimes shallow water can be pretty fast in the times of weeks, or that water could be down there for 10,000 years before it comes back up and we use it. It's down there, things are moving slow, and it can be there for a while, meaning when we contaminate it, it's going to be hard to get it clean. It's going to be dirty for a while. We need to think about how it's getting contaminated. Next slide. And then again, just to paint a picture, when we talk about groundwater, this is where we're at. We think a lot about rain and rivers and streams and lakes. This is all surface water. But as that water comes down, then, depending on where we're at and the local geology and the lithography, then we'll hit water table. And that means then how far do I have to go down until I hit water? How far do I have to dig my well before I get to a good water source? And what it's doing is there's a water table, and there's cracks and there's fissures and there's water moving through all of that. So this is what groundwater is. And even when you get down to me as a microbiologist, why am I studying this? Look at this lower right-hand side or lower left-hand graphic here. When you get to the micro scale, or where these get to particles and sediments. I mean, these are millimeters to micrometers, and this is what the water is moving through. There's water and there's gas, but there's also microbes growing as biofilm on the surface of these sediments, and they're actually helping to clean the water and filter it out, take out contaminants and other nutrients that we usually think of clean water not having. So they're part of this system. And this is why then I'm funded to study microbial biofilms in the context of subsurface water. Next slide. Next slide. There's enough contaminated groundwater in the United States right now to fill an aquarium that has a base of the Washington Mall. How many have been to the Washington Mall? And the sides of this aquarium are 7 miles high. That's how much contaminated groundwater we have in the United States. That's not counting the previous USSR, not counting China, not counting India. This is just the United States and the amount of contaminated groundwater that we're dealing with. Next slide. Superfund sites. So Superfund sites were designated in the early 1970s with the advent of the Environmental Protection Agency. Superfund sites are sites that need a Superfund to help clean it, meaning a money pot. So what was happening before we had the EPA, companies would come in, industrial companies, we'd get a lot of contamination. Local communities would get mad, people would get sick. They'd come to the company and say, you need to clean this up. The company would file bankruptcy and say, we're done. Who's left to clean it up? The American taxpayer, me and you. So the EPA then instituted what was called the Superfunds. And so when a company started and they did an environmental impact assessment and they have the potential for impacting the environment, which most do, they have to put money into this fund so that that fund is there to help clean up the messes that we make. And this map's probably a couple years old now, but this is all active Superfund sites across the United States. You can see a lot across the eastern seaboard, in the western seaboard where we have most human population and industrial activity. But on average, nearly 1 in 6 Americans live within 5 kilometers of a Superfund site. We don't realize that. There's a couple Superfund sites, past and present, in Bozeman. So where the library was a Superfund site, right? That was the train depot. There was a lot of chemicals there, but that's been cleaned up, obviously. So there's good ways to use this Superfund site. But we're talking about impacting probably 55 to 60 million US citizens are living within close proximity to these sites and don't even realize it. Next slide. This is one of the sites that I've worked at previously. This is a Hanford site. Hanford is in Washington State, was actually part of the military complex, part of the Department of Energy during the Manhattan Project. They were enriching plutonium for the atomic bombs that were dropped. As you can see, all of this industrial activity is along the Columbia River. Next slide, or push again. So Hanford produced 2/3 of the plutonium for one of the bombs that was used during the Manhattan Project. They had, and I've been out there before, and once you pass the guard towers and the guys with the machine guns, you go out on this high plateau desert and there's these old reactor cores just standing with radioactive signs everywhere. And when they were running those radioactive units to enrich for plutonium, they had to use water to cool those reactors. And they would use chromate, sodium dichromate, to prevent corrosion. And I talked about it in the beginning that we study metal corrosion. These reactor cores, you don't want them corroding and leaking this water everywhere. So they would use sodium dichromate as a microbiocide. Well, what did they do with that water that had the sodium dichromate in it? They just put it into the ground. Next slide. Or one more, yep. And so there's 1.5 billion gallons of chromium-6 contaminated water that went back into the ground. As you can see, you're in close proximity to the Columbia River. What's groundwater do? It wants to flow, and it's making its way to the Columbia River. Next slide. Next slide. I think, yeah, go on. And so there we go. And so what you can do then is you can put wells into the ground. We go into the water table where this aquifer is, and this is where the contamination is. Shown there in the red is, for instance, in this case, where the chromium is. And as water's moving through these wells, then we can actually feed microbes. And we feed some food down there, and we get the microbes to do their activity that they're interested in. In this case, here at the lower equation, you take chromium-6, that's the bad stuff here. Microbes can dump 3 electrons onto it and turn it to chromium-3. It's just a speciation effect. What happens when they do that is chromium-6 is soluble, it moves with the water, it gets everywhere, it gets in drinking water, humans drink it. When you stimulate the microbes and they dump electrons onto it, take it to chromium-3, it precipitates out a solution. It's not soluble anymore. You're not getting rid of this stuff, but at least you're going to have it stay put so it doesn't get into drinking water supplies. So as the water is moving through these wells, we stimulate the microbes, they have this activity, less chromium coming out on the other side, and you stop it from going into the Columbia River. Same thing can happen with uranium, and we do this at another site I'm going to mention. Microbes will take 2 electrons, put it onto U6, and take it to U4. U6 is soluble, moves in the water, microbes reduce it to uranium-4, falls out a solution. Next slide. Next slide. And so this is one of the studies that we've done in the last 2 years where we were trying to actually understand that process, that chromium reduction, going from Chrome 6 to Chrome 3. And it's just, and you don't want to just add lots of food, right? Too much of a good thing is not a good thing. And so you actually have to understand the local geochemistry, what's in the water, what are the microbes doing, what microbes are there, what activity do they have, what kind of food do they like, how much of that food do you add, and you can actually optimize their activity. And so we found by looking at the ratio, in this case, lactate is the food. And if we understand the ratio of how much lactate we add to how much sulfate is there, we can actually, because this is bad for them too. Chromium actually is toxic to microbes as well. And so what you're trying to do is find an optimal case where you can stimulate their activity and they're the happiest that they can be, so they carry that function out that you want. Next slide. Another site that we've worked at is Oak Ridge National Lab. This was Oak Ridge, Tennessee, also part of the Manhattan Project. And this is where they were getting the uranium enrichment. This is the large, and so they enriched uranium. They had 3 ways of doing it. They had uranium contaminated water. They put it into big ponds. Obviously, those ponds leaked. And now we have uranium contamination getting into the groundwater in this area. This is the largest mobile uranium plume in the United States, if not the world. Next slide. And as I mentioned, we can also stimulate the microbes for them to carry out the activity of taking U6 to U4. Oxidized condition, uranium is in the U6 state. Microbes can reduce it, take it to U4, and it falls out of solution. At least it's going to stay put. It's not moving. And we actually did this work now about 10 years ago. And this is just showing a couple figures and papers that we have done. And this was, if you just look at that graph in the top right, those that you're looking at uranium concentrations, mgs per liter going up to 10. And then we slowly started to treat this water, feeding the microbes and understanding the microbes that were there, where's the communities that were there, what was the activity they were doing. And then you could see that took 700 days. It took a couple years. But by the time that red dashed line is actually the EPA drinking water level standard for uranium. So if you could drop it below that line, the EPA says that water is safe to drink. And we were actually able to do that. And this was the first demonstration at a field site in the world where we were actually able to treat the uranium and get it to stay put and have water coming out that was clean. That's that lower middle graph on the bottom, or image, is just what the ponds looked like 50 years ago. And they were just dumping. Basically, when people ask what's in those ponds, I say the periodic table. And that doesn't even count the organics that are there. But you can see the schematic on the lower left is kind of what was done. Put wells in, caused a recirculation loop, and in this area in the field, we could actually drop the uranium level. This was work done on a huge collaborative effort. It took microbiologists and geochemists, environmental engineers, and civil engineers. A lot of different people with different expertises to actually go to the field and apply this type of research. Next slide. So now we're still at this site, and we're trying to then study the microbes that are there and understand them better, and how they're responding to these contaminants, and how they're responding then to what we're doing to promote them to then do this reduction of uranium. And then how do we, now that we achieved it, but then how do we get it to stay that way? And so that's what one of the areas that we're trying to understand, this idea that I set forth in the beginning, structure-function relationship. How are these microbes structured? How did we change that structure when we went and started feeding them food? And is there a structure that can maximize the function we want? And the function here is uranium reduction. So we're still going in, we're doing long-term studies, trying to understand microbial communities and how they respond to different parameters in this field site: dissolved oxygen, pH, nitrate, sulfate. As I mentioned before, right, when we did that other study, looking at the right ratios of lactate to sulfate, you just can't go anywhere at any field site and just dump some goodies in the ground and get microbes to do what you want them to do magically. You have to kind of understand what microbes are there, what are the surrounding conditions, What are the parameters you're trying to control? And then how do you control those parameters to get to happen what you want to happen? Next slide. Next slide. So this would just be, I'm not going to go into detail here. This is just an example then of how we start to use DNA sequencing to track microbial communities over time and space so that we can understand what microbes are there, right? When I talk about looking at microbes, which ones are there and their populations, right? We have to do that all by sequencing. Most microbes look all alike. They're either little balls or little rods, maybe a curved rod, maybe a spiral. That's it when you talk about morphology of a microbe, and they're all tiny. It's hard to see to begin with, right? So what we do really is take lots of samples over time, we extract nucleic acid, and then we sequence that DNA, and we can tell then what microbes are there just through their DNA signatures. And then this is looking at population distributions over time and space and understanding what microbes are where. And that's kind of how we begin to do that type of work. Next slide. And then we start to relate that. These are models of population distributions for microbes related to the geochemistry that's at the site over time and space. And we start to then make these, we map these out and try to understand, bring some sense to these hairballs of what's going on in these relations. These are quite complex. There's lots of microbes there, thousands. There's lots of parameters going on. They're all related to one another. And so this is what when in the very beginning, when I said causal, this is why it's such a big question and why it's so hard. How the hell are we supposed to know what the cause and effect is here? And that's been a challenge across microbial ecology, irrespective of what you're studying. Water, dirt, plant roots, human gut, it's very difficult because microbial communities are so complex, very heterogeneous, changing a lot over time and space. What are they responding to? That's been the hard part. Next slide. But we're trying to do that, and this was one of my students in one of her papers last year, where what we've done is we've taken some modeling that's used quite commonly in economics. So economics is another parameter, another example where you have lots of variables coming in. Everything is codependent, but yet you do have cause and effect. And so I challenged my student to think in a way to take our DNA sequencing data, but use these Granger causality models that are used in economics, and can we start to make predictions, cause and effect? And these are very, very simple, but it's a different way to start with it. And so, for example, what this graph is saying, go to the lower portion. FW303 is one of our wells, and we're looking at species richness. How many different microbes are present in this well at any given time in the groundwater? And we started to make a model and said, okay, when oxidative reduction potential, the parameter we can measure, it tells us something about the redox of the environment, how oxidized or reduced it is. When it changes, and we measure that change, we predict, this model says, 4 days later, we're going to see an increase in species richness. So the microbial community is responding 4 days later, and we'll have more species there when we see an increase in that ORP. So this is what I mean by getting to, and this is very simple, and it's probably wrong. Most models are wrong, but they help us ask questions, right? And poke a stick at the system. And so this lets us make a hypothesis. Okay, when ORP goes up, does species richness go up? Now we can go to the field and actually see, does that really happen? How good is our model working? So that we're starting to work towards that. Next slide. Another area that we study is actually coal seams in eastern Montana. Montana holds, I think 6 to 7% of the world's coal reserves are actually in this part of the country. And with a lot of these coal seams, they're too deep to mine, but they make a lot of natural gas. And so people want to understand how that natural gas is made. Are microbes involved? Yes, they are. That's why we're there. And can we stimulate them to make more natural gas so that we can harvest it? And or can we just understand the turnover of recalcitrant carbon source in the subsurface and how that's making a greenhouse gas? Understanding that process just in a fundamental way. And so we have a study site that we work with the USGS. These are different pictures of us working out at that site. It's outside Boerne, Montana. Next slide. And these are just, again, we're trying to understand the microbial communities that are down there. What field parameters are having a big impact on their ability to turn over the coal. They're actually chewing on this coal very slowly. Those byproducts get cross-fed to methanogens. Methanogens are microbes, the only ones on the planet that can actually make methane. This is what we mean by biogenic methane. That methane's being made by methanogenic archaea. And so we track all of these populations, and we're trying to understand then the field parameters that control the turnover of this coal and how it gets turned into natural gas. And another example then of structure function. How are these microbial communities structured in this given subsurface environment, and how does that impact their function? In this case, the function is natural gas production. Next slide. And these are the last few examples of work in my lab. I mentioned we'd look at algae and diatoms. And we're interested in growing algae and diatoms. Let me see. Can you advance? I think I'm maybe a little bit more. This is a good one. And so algae and diatoms have the ability to make, so like if I start, as I've gotten older, I've noticed I'm sort of getting carbon reserves. And what do humans store carbon as, for better or for worse? Lipids, fats. So do algae and diatoms. They do it as an oil though. And so what you're looking at are small algae and diatom cells. And that gold or yellow gold color is actually tiny oil droplets. And under certain growth conditions, they will store excess carbon. That's their fat reserve. But now it's an oil droplet. You can harvest these cells, bust them open, and get that oil. And it's no different than the oil that we could go get out of the ground right now. And we can turn that into biodiesel, or we can turn that into other things. And so other professors here at MSU, we look at algae and diatoms, how you eat CO2 out of the air, how you can use waste nutrients, wastewater, and then get these algae to make oil for you. We're interested in, obviously, in biofuel, one way, but we're also interested in these algae because then we can use that oil for fish feed, for aquaculture. So the two different reasons that we're interested in growing these algae. So go back 2 slides. Yep. And so this was one paper that I think was published last year, a student in my group, where we looked at the wastewater treatment facility in Logan, Utah, one of the largest wastewater lagoon systems in the country. We got excess nitrogen and phosphorus. We're trying to get rid of it. Algae like to eat nitrate and phosphorus. Are there algae in here growing? And it's obviously a very diverse community. And do they make oil? Next slide. I'm sorry, forward. Forward, yeah, the other way. Nope. There we go. Yep, keep going. Keep going. And so we just sampled over time in these different lagoon systems. And what's being shown here really is we're looking at their fatty acid methyl esters. That's really what you're going to turn biodiesel in to burn in your vehicle. And we take that biomass and turn it into FAMES. And yes, it was there, and it's higher and lower at different times. And so we're trying to understand the community, the microbial community in these wastewater lagoon systems and their ability to then make oil that we could harvest at different times. That water's just sitting there, not being right. We're trying to clean it up before it's released. Nothing's happening with that biomass. Maybe we can start to couple these processes. And as we're treating wastewater to get rid of waste, could we in turn then be getting biofuel out of it? Next slide. And again, we're looking at the microbial communities that are quite diverse here. So for instance, we're interested in, if you go up there to the sort of in the center, a little bit up above the center, you see a chlorophyta. Those are the green algae. Those are some of the organisms that are making those lipids. What populations are they connected to? And black lines then are positive relationships, and red lines are negative relationships. And so those populations would be higher at certain times and lower at certain times when there's other microbial populations present. So then trying to understand this complex community to try to optimize it to be getting to a point where could we get it to make more oil in a certain way than other times? Or can we tweak the way that we're treating our water so that not only can we get clean water, but we can also get biofuel out of it as well. Next slide. So I hope you, and again, I mentioned that in the field site, it took people, professors and researchers and scientists and engineers from lots of different fields. So I've had in all the things that I do, right? I'm doing microbiology, but it's biochemistry and it's ecology, it's geochemistry, it's statistics. You always need a statistician no matter what, with the way that data is working now. Bioinformatics, physical chemistry, and as I mentioned, engineering. And so you'll get to, it's important that you got to pick an expertise and sort of dial into your area, but always be open into working with people from other fields. And I've been fortunate to be able to do that and to apply the types of work that I do. Next slide. And so at the end, I'll just mention, it's always going to be big data, because as you can see from these types of things that I get to do, I work with lots of different folks, lots of different people from different fields, lots of different types of data. Big data is here. It's going to be the way that we work. It's going to be the way that we do things. Data science is going to be huge in how we use that information. I don't know if you know Carl Sagan, he's a famous astronomer, space scientist, and he said, right, we as a society are becoming increasingly dependent upon science and technology, but we have fewer people that understand science and technology. And so you need to be able to go out, have an understanding of these different fields, and be able to work with people, and you'll be the one then helping to make decisions. You'll be the ones that are going to go out and try to solve some of these problems. Next slide. My slide, this is an older slide from last year of my group. I didn't get to update it. But these are folks that have been in my group probably for the last year or so. Some of them have left, I have a couple new ones. And as I mentioned, funded by the Department of Defense, Department of Energy, as well as the National Science Foundation. I'd be happy to take any questions. We'll be doing it for a long time. Well, one is because I think it's going to be a long endeavor, and two, we keep contaminating water. We find ways to do that. I wish probably what we ought to really do is be more proactive about as we're cleaning water, how do we be more proactive and stop contaminating the water? We tend to just rush in and do things, and then as a society and as a government, be more proactive and stop contaminating what we got, I think, and the other part of that, I would say, so long time, but I think probably where the payoff will be is to stop thinking more and stop contaminating what we have, and finding ways to keep it clean. The other big thing, if anything, from all of these topics that I study is to stop thinking about ourselves, and I include myself in this, obviously, as a one-time use society, right? We use something, we throw it away. And we've got to think about reusing things. Be it, our water, but even think about, we're all carrying laptops and cell phones, and think of the rare earth elements and heavy metals that are in all of these motherboards, and then we just throw them away. We've got to get that metal back out, not leave it in the environment, and find ways to reuse it.
I guess I could make a few comments. I've told you guys in the past that the last century was a century of chemistry and physics. You can see a lot of chemistry and physics, and you can see how biology was ignored for a long time. And we're still ignoring a lot. For instance, what % of the organisms in the soil can you culture? Because if you can't culture them, it's really hard to tell anything about what they are.
Yeah.
So what %?
Well, 95.
95.
We'll say,
okay.
I'm sorry, 5%. 95% we cannot culture. So we don't know what they're doing, right? That's a treasure trove of biochemistry that we don't know how it's working. Even thinking we do some other work that, as an example, touching upon what Professor Sands is saying and I didn't talk about, we go into Yellowstone and we look at novel organisms there, as a lot of people do here at MSU. And we identified using genomics, we're interested in methane and carbon cycling. And we're finding new organisms that we know are out there that look like they're processing methane or making methane that we don't even know, we can't grow them. We didn't even know they were there to begin with a couple years ago. So we're thinking about, so now back off and think about we're trying to write a carbon cycle for our planet, and we don't even have all the pieces. Okay, so we're trying to put a puzzle. Imagine what we're doing here. We're trying to put a puzzle together. You don't have all the pieces, and you don't even have a picture to look at. So imagine that's what we're trying to do and how we're trying to function.
I find that biologists are really lousy at calculus and general math like that, but very good at interrelations between studying the relationships of different things. And so it's going to be interesting to see where this field goes. I mean,
yeah. And you made the mention of chemistry and physics. You know, the last couple of centuries have really been about chemistry and physics, and there's a lot of laws, a lot of theorems, and we've got what we think is a pretty good understanding for chemistry and physics, and they're very quantitative. Biology inherently has not been quantitative. And so people are really trying, I think here in the next century, people are trying to push quantitative understanding in biology. And that's hard because of the variability. Even on an organism that we do know, because of the variability and the heterogeneity. And that's inherent to a biological system. It's really hard to deal with. But we are finding ways to apply math to that and to get it more quantitative. So regardless of your interest, and I'm going to tell you the same things that I tell my kids. I have 2 kids in college right now. Do all the math you can, regardless of what you're going to do, because you can then bring that quantitative nature to whatever your interest is. Psychology, sociology, I don't care. Do bring math to it. Because again, this notion of data science and big data, you're going to have a way to handle information. And math and statistics is how we do that. So whatever you do, if at all possible, bring math into it. You're going to have to know how to handle data and analyze data. Can I tell one quick story about
you can tell 3 or 4.
About Netflix.
Go for it.
Does everybody know? So here's an example of where you would use math and data in a way that you may not predict. So who watched House of Cards? Anybody would like House of Cards on Netflix? And is it already that? I guess there's just so many new shows that come out. It's like, it's an old scene. When did House of Cards come out? I don't know. 7 or 8 years ago. And what Netflix produced this, and it was just when Netflix was starting to produce their own shows, right? We had these streaming services starting to make their own content. That started to happen. So what did Netflix do? They said, we're sitting on tons of data. I know what millions of people are watching at any given time, what they watch, how they watch it, and they had all of this data. So they started to ask. They said, okay, what type of show do people like to watch the most? And they didn't have to send out a survey. They had it. They had that data. And it was political thriller. And then they said, okay, what actor gets watched the most? And at that time, it was Kevin Spacey. And they said, what director gets watched the most? And I forget who the director was, and it was some director. And they said, how do people like to watch the most? And they like to binge watch. They want to watch it all at once. So they took those 4 things and they said, okay, we're going to make a show. We're going to make a political thriller. We're going to put Kevin Spacey in it. We're going to get this person to direct it. And we're going to release it a season at a time. It had never been done before. And what show came out of that? House of Cards. And it was a huge blockbuster hit. And it's because they took immense amount of data from millions of individuals. And they could analyze that data and ask the right question and had the answer, and they made, I don't know, hundreds of millions of dollars off of that show. And then it's just exploded, right? Now everybody's doing this. So
So-
another interesting part of, we all have laptops and phones, and I try to limit on my phone, like actually social networks and stuff. But when you're signed into all those things, and you're looking, every time you look at something, or you click something, or you say something, or you go somewhere and do it, they're tracking you. You're giving that data up. Just think about that, because those companies are tracking it, and that's how they're making decisions. And then they're coming back to you, putting stuff in front of you, and impacting decisions you make. But that, I digress. That's another topic.
So in your years of work, have you noticed that the data collection gets cheaper or easier or automated? Is that part of the big data, or is it still like a lot of work for you and colleagues to go out and collect this?
I think comparing it to like social network platforms, right? That data is just so easy to get, and they can write the algorithms that are just underlying everything and taking that data. For me, doing these experiments, it is always still a limitation, right? We know there's all these things that we should be monitoring, but it costs money and time to get them. And so I still think, but once we get that data and like housing it and storing it's getting cheaper, and actually the analytics to go through that data is getting easier. The bottleneck is still getting the data, in the sense for me as an experimental biologist and working with engineers, the cost is still getting that data. Now, as what happens, right, is we're thinking about automation. So then maybe we've got drones, and maybe I'm going to fly a drone and I'm going to put some detector on it, and now I can go fly it and I can go make gas samples a lot easier. Or I can stick we build things that go down the wells that then have the ability to do telemetry and just send data back. So I don't have to go and grab a sample, bring it back to the lab, do the analysis. If we can then build miniature things that go down the well and just do it in real time and make the measurement, that's where the impact is. There's still a lot of research to be done, but that's where automation is going to come in and impact my ability as a biologist then to make measurements, because then we will be able to make more measurements over time and space, more data. Now, then we're going to start having big data.
So put on your cell biologist cap for a minute. And they all know about computers. Could you compare a cell and the decisions it has to make with a computer and the decisions it has to make? That's a tough question, but that's an interesting question.
Well, and I used to teach, like for the first 10 years that I was here, I taught microbial physiology, which was a 400, 500 level, basically bacterial biochemistry class. And we would talk about biochemistry and the physiology that they're doing. And we would talk about electron transfer and making ATP and running the Krebs cycle and glycolysis and then the Dudoroff and processing carbon and nitrogen and all these things. And we talk a little bit about genetics, and I would always make the analogy of, you have this single cell, it is looking at all these environmental parameters and assessing them and making decisions, but there's no computer board. There's no algorithm in the way that we think about an algorithm, that we write a code to do something, but yet this is what they're doing. And so in a lot of the ways they're functioning, they have sort of this inherent way to mimic their environment and make decisions, but it's not with computers and such. And so they're doing these things. And so we have a lot to learn there. And I think it will change for better or for worse, right? When we think about artificial intelligence and we think about the way that we're going to build computers and what they do, we're probably going to start to learn more about how not only just our brains work, but a single cell works and how it makes decisions. And we're going to incorporate that into these intelligent things that we're building, for better or for worse. There's a lot. That's a really big topic, and there's a lot going on there. But I do believe, right, and even for a microbial cell, I would tell class, right, microbial cell is continually dipping its toe in the pool. Continually. 5, 10 second time scale, right? And it's looking at what's out there. Okay, what sugar's out there? What nitrogen source? What phosphorus source? Who's around me? And it's continually then deciding what genes to turn on and off. And then it picks a set of genes to turn on or off. Then they're transcribed. Then they're translated. Then the protein's there. And then you have an outcome. And that's happening on a half time of seconds to minutes. And then it starts all over again. But yet it's very simple. And so I tend in a very similar way, I tend to think about this notion of biology and how it works. And it's this notion of simple complexity. We know they're complex and we know there's all these things going on and they're processing all of these variables, yet they do it in a very simplified way that we don't understand. That was probably a long answer to that question.
So if we take an organism like E. coli and it has 3,000 genes, and we look at which genes are expressed, it's about 10% of them. And your neighbor may be doing something so you don't have to do it. If they make alanine, you don't have to make alanine. So this whole community thing, even within cells of the same kind, okay? And in fact, you're looking at what we call a multicellular organism here. If you take organisms out of my cheek and you look at one cell, I don't mean organisms, I mean cells, they're cross-feeding. If I make something, you don't have to. And maybe that's why multicellular organisms are so successful in a way.
No, and that's another big question of microbial ecology in general, is this notion of metabolic interactions and sharing, and it's probably much more common than we realize. And it's not that microbes have made this decision on like, okay, I like this guy and I don't like this guy. I'm going to work with you and I'm not going to work with you. It's really at a very simplified level of what's in my environment, and some guy happened to make alanine, let's say, and then I can make alanine, and then I don't have to turn on the genes to biosynthesize alanine from scratch because I can just bring it in. But I happen to be making arginine, and so then I make a lot of it and it's out in the environment, and that guy doesn't have to make arginine. They're not making a decision, a high-level decision to say, okay, let's swap these alanine and arginine, but it just happens through mass balance. And again, this notion of simplified complexity.
There was a guy named Adam Smith. He was an economist, and he said the most efficient way in a village is for one guy to make horseshoes, not everybody to make horseshoes. Another guy would make the nails, and so on. And so I think we're starting to see this kind of community interaction in organisms. We have a long way to go, though.
We have a long way to go, and I've heard it said in some papers, at least, that we always get hit so hard, right? The survival of the fittest. It might be survival of the friendliest.
Thank you.
All right, thanks, guys.
Discussion at the end.
Yeah.